Hi, I'm Dr. Ahmad with the Department of Surgical Oncology at Stanford University, postdoctoral research fellow. I am presenting on a project. It's a single-cell analysis of melanoma that revealed a myeloid cell signature associated with survival in early and advanced melanoma and a response to immunotherapy in advanced melanoma. So briefly, we sought to kind of figure out if there was a myeloid subtype that would characterize biology and support therapeutic strategies in melanoma patients with diverse biogeographies. And by diverse biogeographies, we mean that we collected data from primary melanomas, primary acral melanomas, melanoma metastases in the lymph nodes, the abdominal organs, and the brain. Methodologically, we combined 6 data sets in 126 patients that had single-cell RNA sequencing conducted of tumors. After integration using kind of the best-in-class pipelines that are available today and that are published, we then clustered using an algorithm that allowed us to kind of make the data set a little bit smaller using our artificial neural network several times, allowing us to run kind of a complex clustering algorithm called k-means analysis that usually requires a lot more processing power on a laptop computer. And so by allowing us to do that, we were able to cluster, several hundred times or more in order to find kind of the optimal number of groups, which led us to find new groups of cells or new types of cells that may be present in the tumor, especially in rare cell types. And that's what we were really trying to look for, rare cell types that haven't been discovered before. Once we were able to figure out all the different cell types that were in the tumor, using kind of biology annotation and our clustering analysis, we then conducted recursive partitioning analysis on the different cell types based on an outcome of survival in our training data set. So of these 126 patients, there was a little more than 40 that had brain metastases and had survival data. And so with those patients, we conducted kind of a training analysis using RPA. We're able to find one cell type that was prognostic with survival. And then, did differential gene expression analysis on that particular cell type, made some Kaplan-Meier survival curves, and then did a pseudobulk comparison for that data set. We then validated our signature using an approach called deconvolution in 2 bulk RNA DNA sets. And so those DNA sets were from 1, a clinical trial, and 2 from the TCGA. So I will get into the results a bit here and show you a bit more of the figures. And as we zoom in here, you can see here's about 100,000 cells from 126 patients. And this is just kind of a figure representing what all of our cells look like plotted on a 2-axis plot. Over here are all the different myeloid cells we found. So we found about 6 different cell types. And this is what our RPA analysis was. And we just found this was a cutoff score of 2.6% of those cells in the entire tumor were of the cell type. We're calling it right now MC1, or myeloid cell type 1, which requires a name kind of as we move forward into the future. And of the cell type, if it was above this cutoff score, and then our survival was very good. As you can see here, the survival was 52 months since getting a diagnosis versus 32 months in the middle and 13 months in the lowest group here. And that was all based on a cutoff score based on percentage of these cells in the tumor. This analysis here, volcano plot, shows us that, definitely there, if you zoom in a little bit here, you can see that we identified that this was not a melanoma type of cell. This was clearly a other type of immune cell. And so a lot of MHC class 2 genes are expressed highly. And then our baseline characteristics analysis, are there any treatment differences that could explain this potentially? We did find that there was one significant difference on post-biopsy treatment, and it looked like targeted therapy was increased in the best surviving group, but also the lowest surviving group. And so it didn't quite make sense when we compare, the top group, we call this node 2 to node 4, which is the middle, as node 4 had better survival than the node 5, where more people received targeted therapy. So unclear how treatment would affect this cell infiltration type. And so we sought to kind of take this finding and validate it externally a bit more. So we were able to get access to some clinical trial data that was published for a drug called nivolumab. And so these patients had never received the drug before. During this trial, they had a biopsy before treatment. They had a biopsy of the tumor while they were on treatment. And then the patients were followed to their endpoint, which was either progression or survival. So you can see here, again, we repeated the cutoff scores. So we use the same percentage scores that we calculated using our training data set. And this is overall survival plotted by the infiltration of this MC1 cell and the on-treatment biopsy. And you can see here in the highest surviving group and the middle surviving group, these again are significant. But when you compare this to the lowest surviving group, we can see that this node 5 is no longer kind of the lowest surviving group. So that was interesting for us. However, if you compare kind of node 2 and node 4, here on multivariate regression or Cox analysis, you can see that it is still significantly different. And the hazard ratio is 2.35. So patients with a low score or low percentage infiltration of the cell type, have almost 2.5 times the risk of dying. And so that equates to a survival of 28 months versus 12 months, which is pretty significant. That's almost another year of life. And you can see here that node 5 really didn't have very many patients in it all. Had one patient in our data set here. And if we looked at baseline characteristics of these patients related to cell infiltration, that shows us that these are significantly different. And then we also compared it to the standard kind of test, which is overall response based on changes in tumor size. And so we did see some standard differences. So between 2 and 4, we saw that there was more progressive disease, 64%, and node 4, which is the lower surviving group compared to node 2. However, you can see that complete response was about the same in both groups. And so there is a difference between CT response and the response we calculated based on tumor infiltration of our cell type. And we kind of have to get more detailed information on why that's the case, why we may have some different results than what you see on CT scan or CT score based on RECIST scoring. However, we did potentially also show a little bit better prognostication and survival when compared to CT response alone. Then we decided to externally validate the model once more in the TCGA data set down here in figure 3. And so we took all the data that was available, used deconvolution, and then used the same cutoff scores again. So there's the high group blue, the middle group red, and the low surviving group green. Green actually had an infiltration score between the highest and lowest scores. And you can see here that kind of lines up with the highest infiltration score. So again, there wasn't a big difference between those two groups, but between these two groups, the high surviving and the low surviving group, or the high infiltration versus the low infiltration cutoff, where most of the patients again fell, we did see a significant difference. And you can see here it's about 2 times the risk of death in node 4 versus node 2 with a P that's very statistically significant. Now, what was interesting about this data set is that we were prognosticating patients in earlier stages of cancer, so stage 1, 2, and 3, and including some stage 4 patients as well. And so when you can see here that this number of stage 4 patients from this data set was much smaller based on when the tumor biopsy or the treatment tumor biopsy and sequencing were done. And so in this case, we were able to actually prognosticate patients in stage 1, 2, and 3 groups, which is very early. And so what's interesting here, and all the survival curves aren't presented in this particular abstract in this current presentation, but what we did see is that we were able to tell the difference between patients and their survival, even in stage 1, which is very, very early and would be a better understanding of cancer at that point, because at this current time, there's really no test that tells us whether someone will recur early with essentially potentially a curable stage of cancer like stage 1 melanoma. So that made our finding very interesting and requires that we kind of look a little deeper at it. Our conclusions based on our research were that the increased infiltration of this myeloid cell showed improved survival in melanoma, and that include melanoma with brain metastases, advanced melanoma, and even early stage melanoma in stage 1, 2, and 3. In both the training and validation data sets. And then also, curiously, cell infiltration was associated with response to a novel drug or a new drug that's being used more often now in melanoma patients called nivolumab. So it was associated with drug response as well. So what's interesting is that our biomarker is really just a novel cell type. And so this cell type could be used for therapy in the future as kind of a treatment on its own. And so our finding requires further development in order to validate its significance as a potential cell therapy, although the value as a biomarker is exceptional, and we were very surprised with what we found. In the future, we have several use cases, and we have filed a provisional patent on this finding as a biomarker for melanoma. And so it could be used for prognostication in all stages of melanoma, in stage 1, 2, 3, and 4. As kind of I mentioned before, it could help with stage 1 surveillance. So if someone had a resection of a very early stage melanoma, this test could provide them with further information about, being more aggressive on screening versus someone that may not recur, as early as a patient with a low infiltration score of this cell type. And then, it could also help us with treatment selection for patients in the future. If they have a low infiltration score, even in early stages like 2, or more advanced stages like 3 and 4, then we may consider more aggressive therapies at an earlier stage for these patients. In addition, it may help us figure out which patients are responding to a drug while they're on the drug itself. And so that could be helpful in order to help facilitate additional therapies rather than wait for response on a CT scan or a PET scan, or potentially even waiting until, the patient advances in their cancer. And so we may potentially, consider more aggressive therapies or cell therapies for these patients where we, know very early whether they're responding to a drug or treatment before we have any real physical signs or imaging findings that allow us to figure out if this patient is responding to the drug. So there's definitely a lot to learn and glean from our finding, and we're very happy to share it and continue sharing our findings in this very interesting research project. Thank you for your time.